Electrical Engineering, Ophthalmology, and the Graduate School of Education. He directs Stanford's Center for Cognitive and Neurobiological Imaging at MRI Service Center and was Deputy Director of the Wu Tsai Neurosciences Institute from 2013 to 2021. Dr. Wandell earned his undergraduate degree in mathematics and psychology from the University of Michigan. He then moved here to the University of California, Irvine, where he received his Ph.D. in social science. Dr. Wandell then completed a postdoctoral fellowship at the University of Pennsylvania. In 1979, he joined the faculty of Stanford University, where he has been conducting his research up to the present day. Dr. Wandell's research centers on vision science, spanning topics from visual disorders, reading development in children, to digital imaging devices and algorithms for both magnetic resonance imaging and digital imaging. Dr. Wandell's work in visual neuroscience uses functional, structural, and quantitative MRI, along with behavior testing and modeling to understand the action of the visual portions of the brain. His lab has worked to identify and then understand the organization of the visual field maps in the human brain, color and motion processing within these maps, the potential for reorganization following injury, and the development of cortical circuitry for reading. The Wandell Lab develops software tools for digital imaging applications. The software includes methods for analyzing magnetic resonance imaging data, as well as tools to design and evaluate cameras used in a range of applications. His work has resulted in multiple publications in prestigious journals and has led to the commercial applications, including 2 companies that he co-founded. He was elected to the National Academy of Sciences in 2003 and most recently received the prestigious Proctor Medal from ARVO in 2021. We are very excited to host Dr. Wandell here in person today, and please join me in welcoming him and presenting his talk today, Cortical Circuitry for Vision and Reading.
Well, I hope you can all, oh good, we've got a mic here. I hope you can all hear me. Thank you very much for the invitation. It's really nice to come back to Irvine. It's amazing, I'm looking at so many young people. It's amazing you can't imagine what it was like when I was here in 1974. And it was incredibly, it's gorgeous now. It was actually quite gorgeous, just in a completely different way then, with just fields everywhere and emptiness. And you've filled it up with some really brilliant people and some wonderful institutions. And it's a really pleasure to see how it's developed and what's come here. So I chose a couple of things, as you mentioned kindly in your introduction. I'm old, and there's a bunch of things that I could have chosen to speak about over the year, work over the years. And I've chosen because I'm so fond of my colleagues in ophthalmology. I've tried to choose some things that are related to issues that may come up in the field of ophthalmology. And in particular, I'm going to focus on cortical plasticity during development and cortical plasticity in adulthood, which took a fair amount of time for me thinking about it. And this is because in ophthalmology, you're so fantastic at doing work at the front end of the system and wondering what's going to happen at the back end after long periods of problems at the front end is something I think that we, as a scientist and not a practicing clinician or doctor, it's something that maybe I can help with, or at least help you get prepared for as you develop these fantastic devices. I think my friend, I don't see him on here, but Daniel Palanker, hey Daniel, good to see you. Maybe we should be having coffee instead of this though. Working on these devices, working on devices to implant in the front end, or the different kinds of gene therapies that are evolving and so forth, mean that you want to know what's happening at the back end. So a lot of the stories today will be about that. So I'm also going to, let me take you through, let's see if we can't get this to go forward. So as I was just saying, plasticity and stability human cortex is really what the issue is. By the way, sometimes you'll find people, and they may come through here, that'll just say, "Oh, it's so plastic, it's so plastic," and I'm not one of those folks. There's both plasticity and stability, and it depends where and when and so forth. And I'm not going to be one who just says it's amazing and plastic. We're going to see it in other contexts. So. Let's try to figure out when it's plastic, when it's stable. And then I'll take you to bits about how we tried to do something at the intersection of education and seeing and a field that I hope that ophthalmologists might get a little more involved in, pertaining to how it is we recognize objects and see words and have visual acuity. Okay, so let me go back to the beginning. I'm going to do some of the basics first, because in my experience, you may all be terribly expert at this, but my guess for most ophthalmology visits is that the cortex isn't really what you wake up in the morning and think about. So let me just set the stage. So the history of how we figured out things about the organization of visual cortex is really quite amazing. Perhaps the most famous name isn't shown here. It's a guy named David Ferrier. And Ferrier, when Hubel and Wiesel gave the Ferrier lectures, and he's a very famous name. And Ferrier, in this particular deal, is the guy who stood up there and said, "You know where visual cortex is? It's in the parietal lobe. If you lesion the parietal lobe, you'll be blind." And that was just wrong. That was just wrong. But he's very famous, and he did other things that were better. But he got that one wrong. And the guy shown here, Monk, said, "Yeah, no, that's not right." But he did it in such a rude way. He also did lesion experiments. And he said, "No, the actual visual cortex is the occipital lobe." Did it in such a rude way that William James stood up and said, "What is wrong with all these vision people? They're so argumentative. And what are they doing there?" In any event, Monk basically got it right, identified the occipital lobe. It was then Henshin, who's a Swedish neurologist, who studied a lot of people with strokes, and noticed that when the stroke occurred on the dorsal side, the upper side of the brain, the loss would be in the lower visual field, and the lower side of the calcarine, it would be conversely, it would be in the upper field. So Henshin was the first one to see that there was a little bit of retinotopic organization. Certain parts would you lose one part of the visual field, other parts of the brain you'd lose a different part. It was then Inoue working with, I'm showing up here these helmets and rifles from the Russian-Japanese War in 1904 or something like this. And these were damaging instruments, but they didn't kill you. And a bullet might go pass through your head, and you wouldn't die from it, but it would lesion a part of cortex. And he had a lot of people that he studied with this, and he could see even more beyond Henshin to the visual field map, which Holmes had the same situation, but in the First World War. And the Holmes map became quite famous. And actually, if you don't mind, I don't often see this many ophthalmology folks at the same time. How many of you are taught the Holmes visual field map as part of your training? I'm not, just so you know, Daniel, I'm not seeing a lot of hands go up here. So, and I don't think that, I think the same thing would be true at Stanford, by the way. So it's the same story. Nonetheless, it's visual cortex, and the map is shown here. This is a little bit, you'll see a lot of brains coming up, but this is the occipital pole, the very posterior part here. The foveal region is represented over a very large expanse. So it's very small. It's just maybe, 600, 900 microns across on the retina, but it spreads out quite far, over a centimeter in the area V1. And then you can see the representation here. The lower visual field shows up on the upper bank of this calcarine sulcus, which is here. And the upper part of the visual field is represented in the lower part of the calcarine, just as Henshin had told us. So this has then became, it was kind of a quiet period until the start of the electrophysiology revolution in the 30s and 40s. At which point, all the neuroscientists, this sort of the origins of neuroscience in the visual cortex began. And from about the 40s to the 90s, was a massive number of measurements where we discovered, gee, there's not just one such representation, but there's many such maps like this. And I'll show you pictures of them coming up. But that was all in animal models. And when it came to human, there had been, we can't do electrophysiology in human. And so we had this, I could pull out this very nice quote from Jonathan Horton and Bill Hoyt, who are wonderful neuro-ophthalmologists at UCSF, said basically we don't know anything about the second visual map in human in 1991. That's kind of where we stood. And the reason was we had technology that looked like this, PET. And PET is pretty good for some stuff, but in terms of spatial resolution, the ability to measure a lot, this is what would get you a Nature paper in 1985. You'd take a bunch of people and put them in a PET scanner and have them stare at something in the fovea and then keep fixation, but put the stimulus a little bit peripheral. And you'd see that this little spot here would move from the very back of the brain a little bit forward. You're averaging many people. You put a little outline of the brain there. You'd write for 3 or 4 pages, and there you go. You got yourself a Nature paper. And that was the best we could do in those days. But the revolution in functional magnetic resonance imaging, the ability to measure repeatedly in the same person at higher resolution, meant that by about 20 years later, we were able to make measurements of the human visual cortex. And what's shown here, again, is the same posterior, the occipital lobe over here. And the spatial resolution had changed from voxel sizes this big, a chunk of brain this big, to a chunk of brain this big, shown to scale. So a massive increase. And you could put people in again and again to increase the signal-to-noise ratio so that we could literally identify where area V1 is, and then the second visual map, V2, surrounds it. Over here, I'll show you how we get it in a second. And the third map surrounds that, and so forth. And we could keep going, and we did for quite a number of years. You could find out not just the maps, but notice that for these 3 maps here, 1, 2, and 3, on the medial surface of the left occipital brain. Am I okay if I say that? Medial surface, left occipital lobe. You can see that we can measure not just where the maps are, but the eccentricity representation, just the way Holmes and Inoue and Henshin were telling us. And here you can see this is representation from a couple of degrees on out. And you can see the large foveal confluence back here. And notice that the eccentricity maps for all 3, the eccentricity representation for all 3 of these maps is the same. Right? They kind of run together in parallel with one another. The way in which you measure that, we kind of figured this out in the early 90s, was you would have somebody go into the scanner and stare at a fixation. And the first way we used to do things was we would just have like expanding rings like this. And you can literally, I'm running this faster. In the scanner, it takes about 30 seconds for a cycle, but that would be tedious. So we run it a little bit faster here. And you can literally just see the eccentricity map running across all 3 as you go from the posterior occipital pole into the anterior calculus. Notice, by the way, there's a little thing going on here. I'll come back to that in a moment. And it's easy enough to just see the common eccentricity map. And we often represent it. You'll see some maps like this as we go on by literally just saying, when was this part activated? Well, it was activated early. Then that was this fovea. It was activated a little bit later. Then that's the paraphobia and so forth. And then going out to the periphery. Notice that little part I was pointing you to down here. That's another little foveal representation. And it's actually distinct from the main foveal representation of V1. And this has been seen. I'll build on that as we go on a little bit. To find the different field maps, that comes about by just putting in a stimulus that, instead of expanding, just goes round in a circle like this, rotating wedges in the early days. We do it differently now, but this is how we did it back in the day. And you can see that just as Henshin told us, here's the red, here is the lower vertical meridian, green is the horizontal, blue is the upper vertical meridian, and that's a whole hemifield there, the V1 map. Notice that on this side, it's all blue and cyan, maybe a little bit into the green. So that's the upper quarter field. And so it only goes, that far. And that's why we say V2 goes half of it here. And on the other side, where it's now red and green, the other quarter field, that's why we say it wraps it around. And we already knew this from the monkeys, that it was likely to be like this. But the ability to measure it in a living human who might be doing a task, and they might have some disease and so forth, that's pretty cool. And you can do it now in experiments that take about 10 minutes. When we first did it, these experiments would take an hour. Maybe you'd have to do it 2 or 3 times before you got it right. But these days, the technology is just way beyond that. So when Lisa Brewer, who's here, was in my lab, she was also an MD PhD student at Stanford back in the day. And we did a number of things together. And one of the things was we really paid attention. You can see this, we shrunk down the field. So instead of trying to get the whole coverage of 20 degrees, 30 degrees, we shrunk it down so that we would just focus with higher resolution in the central fovea. And that really clarified some things, and particularly clarified that this wasn't a mistake, this separate eccentricity representation down here in the ventral surface. And so here you can see the central 3 degrees are represented just in the posterior and all the mapping together. But there really is a separate, distinct little eccentricity representation right down there on the foveal surface, on the ventral surface of the fovea. And in fact, as we pursued this over the years, and this was probably 6 or 10 years worth of trying this and checking, we and a whole bunch of other groups observed that, yeah, you've got one eccentricity representation here. Here's the second one I was just showing you in the ventral occipital region. That one, by the way, can also be separated into a couple of maps. It's not just one map, but just as we could separate the V1, V2, V3, we could separate the ventral occipital. And then there was another one on the lateral occipital. So, okay. And then there is another one we stuck with the notation. Actually, sometimes I call this temporal occipital, but MT is so famous amongst neuroscience.
Recording in progress.
So does that mean we missed the first part? Could be. And then another couple up here along the intraparietal sulcus. And for those of you who are students and might do this, all the names that were floating around in the literature, if you go back and read the literature in those days, they all have these weird, monkey physiology names, but we managed to persuade people to wipe that out. And if it's in the ventral occipital, it's called ventral occipital. If it's in the lateral occipital, it's now called lateral occipital, 1 and 2. And for those of you who are students, all I can say is you're welcome. That was a battle, and I'm glad you don't have to go through it. Okay, so the next thing, once you kind of figure out where the maps are, the next thing you might do is try to figure out some computational models of what's in the maps and how are they responding, and why do you have so many maps? And I'm glad to talk about. I won't give an answer to that today, but I spent a lot of time worrying about that and talking about that. But one of the things you can do is the same kind of thing that the physiologists have done for many years, all the way back to Hartline and Kuffler, all the way back to Sherrington. You can look at a particular voxel and you can just start sweeping, run a bar through this visual field and ask, well, what happened at that voxel? And try to map the receptive field of that particular voxel. And so Serge Dumoulin and I started a little industry of quantitative modeling of the response, put different stimuli through there and try to predict what the response would be to any particular stimulus. And we started, as anybody might, with just linear models of what the fields would look like, and that gets you a certain distance. And you can see very big differences. If you look in V1, the spatial receptive field, which is what's shown there, is quite small. If you look in MT regions, the regions on the temporal occipital lobe over here, the receptive fields are much bigger. And in fact, if you look within a map, so the lower one here is V1, as you go from the fovea to the periphery, the size gets bigger. As you look across the maps, that's the different colors over here. The sizes just get systematically bigger across the different maps. So there's a great deal of order into what these signals look like. And then the next thing that particularly Kendrick did, Serge, a number of us, was look at the more and more complex models to explain larger and larger classes of stimuli. And that's an ongoing project, and I'm happy to talk about that computational project. But it's not exactly a ophthalmology thing. So talk to me about that if that kind of computation is of interest to you. So by the time, I don't know when we wrote this, maybe 2008, 2010, something like this, we and a number of others, I should mention Sabine Kastner, Roger Tutel, Marty Serino. There'd been a whole bunch of us competing with one another in a struggle. Who's going to get the next map? Who's going to do this? Who's going to do that next? And so forth. And by about 2008, 2010, that game was over. So we all moved on to the next games. And there had been some agreement. At least 2 or 3 groups had seen something like 16 different visual field maps spread across the brain. And there's now just kind of a big question of how they're organized and what they're for and so forth. And I'm not going to go in that direction today, but I think it's a great topic for basic science research, and particularly relating it to all the new developments in computational vision. The direction I am going to go is at that same time, the wonderful Nobel laureates for the field, Hubel and Wiesel, were looking at developmental plasticity. And that was, an important thought. And Mike Merzenich was up at UCSF saying, "Oh, the brain, everything is plastic, everything is plastic. My God, the brain hardly stays stable at all." And John Koss was kind of backing up. And I enjoyed all these folks and sort of started to look into it a little bit. And so we started to do some experiments. But then because of communities like yours, I'm sitting at my desk minding my own business one day, and somebody calls me up and says, Brian, Joyce Liao, actually, as an ophthalmologist at Stanford, calls me up and says, "Brian, we have this patient here. It seems a little weird. Would you mind measuring this patient because he doesn't have a chiasm?" Okay, no chiasm. So that's interesting because we could measure the visual field maps, and here's somebody who's miswired at the very front end. So again, this I'm guessing you do all get lectured on. Just you'll tell me that the outputs of the retina, the nasal side always crosses to the other hemisphere, and the temporal side stays on the same, and that's on a typical subject. And that means that, for example, the right hemisphere sees the left visual field, the left hemisphere sees the right visual field. And if you don't, and that's what it looks like in an MR scan, easy to see. And this is what this guy looked like, okay? No chiasm. And there's a molecule that in fact signals that that the nasal axons from the optic nerve are supposed to cross at the chiasm. And if you don't have that molecule, they don't cross. And now all of a sudden you have like one eye goes straight up to the same side. The other eye goes straight up, and there's no crossing, no hemifields. Both hemispheres, well, they weren't sure. They called me up and said, "Could you confirm this? Because I don't really trust this MR stuff, really." And so could you confirm what's happening in the map? And sure enough, here's an example of an experiment from the paper. This eye is open, and this eye we covered up, and we put things in the left hemisphere, and it's the right eye. And so sure enough, you get a response over there. But you know that in a typical, if you flip the stimulus to the other side, the response would flip to the other side. But in this guy, you flip the stimulus to the other side, stays on the same side. And you have both maps overlaying one another in the same hemisphere. So let me say about this guy, you might think, wow, this guy must have trouble seeing, right? No trouble seeing. Reads, plays baseball, doesn't have stereo vision, has a little bit of, he came to us because he had nystagmus. And then the nystagmus always had a little, and the nystagmus got worse. But mostly he passed all the standard clinical tests as we describe in the papers and so forth. So this guy is completely miswired in the front end. And yet, so his V1, V2, V3 are completely unlike any of you in this room, and yet he could read, walk around just fine, never really complained about his situation. We actually had two subjects like this. Michael Hoffman, a great collaborator in Germany, had one, and we had one, and we were at the moment we were saying, who's going to go first, and we just said, let's put it all in one paper together, and Michael was a great collaborator for that. So we could then try to measure receptive fields in people and in these people. We did it for the ones at our end. Serge and I had just been developing the PRF, the population receptive field methods back in those days. And the way you run those is you put some stimulus in here and you move these bars through. And you could see that the fits of the models that we would have to apply to explain the data from this particular guy would have two Gaussians, not one. And it's like the retinal representation, sensibly enough, was folded over itself like this and came on up, so that in the world, when you referred it back to the stimulus, he had these symmetric Gaussian receptive fields out there, and the curves are the fits and the points are the data, and then yeah, it was a pretty good explanation of this guy. So one of the things that's interesting about that is that it's a miswiring up front, but everything else sort of stayed the same. In other words, when you looked in V2, V3, and so forth, it continued the bad V1 representation. That didn't change to try to compensate for it and so forth. And yet somehow or other, the ability of the visual system to interpret it just stayed the same. So I've always wondered, there must be a massive amount of ability to learn and to deal with whatever input you have in the other parts of cortex. And the guy did not, by the way, make any right-left confusions, any of the things that you might have just quickly thought of. So that's interesting, and it tells us probably less about the visual system, which just seems stable, than it did about the rest of the cortex's ability to interpret objects and see patterns and judge positions and so forth, given a miswired front end. So I've always thought I should spend more time working on that. Okay, let me say one other thing about this guy. During the years that I had been developing this, I'd also had the good fortune to work with Franco Pestilli, Hiromasa Takamura, and others, and we were spending a lot of time on tractography. So tractography is a method using diffusion-weighted imaging to go and make estimates of the tracts. And I've just given the last 3 lectures in my class, I've been all about tractography, which is mostly filled with the things that it's limited on. But one of the things that it's not limited on is when you know that there's a big tract there, and the fact that you're all looking at me and I can see your eyes and you're following and so forth means that I'm pretty confident you have an optic nerve, an optic tract, and optic radiation. I'm pretty good with that. And we can use diffusion-weighted imaging to go and find them in your brain. So we could do that for this guy who had been miswired, and we could look at the sizes of them. And he was just at the edge. He wasn't off the distribution on anything. So this is, for example, a measure of diffusivity in each of these different regions. The ellipses are showing you the distribution. We had 30 controls sitting around in our databases. And so I put an ellipse of where the distribution is for these values, measures of diffusivity. And he's really not off the charts anywhere here. And if you look at the size of the corpus callosum, because typically you go from between the 2 hemispheres through the corpus callosum in the very back, called the splenium, you can say, well, how wide, how much of an area is there going through the splenium? He's small, corpus callosum, small splenium, but not off the distribution either. So even though there had been this miswiring at the chiasm, which was obvious, there wasn't much of a big effect in any of the wiring of their places. So we were glad to know that. And I have to say, although, and this has now been seen, Michael Hoffman carried on with this research over the years, and there's nothing really to change in the big part of the story, though many details, and I recommend Michael's work and others on this. It turns out he and Irene Gottlieb have found more people like this and use. Okay, so at that time, so that's a case where part of the system is miswired. I wouldn't say that that's developmental, but I would say, you know, it's a miswiring in development, and the brain is quite plastically says, okay, I can deal with this, and moves on. And I had been listening to Mike Merzenich and John Koss and all these others talking about plasticity. And I have this very close friend, Logothetis, who had been given a wonderful opportunity at a Max Planck Institute, and he went and built himself a rig that's shown here on the left to do these experiments in animal models. And in fact, he could put in macaque. And as you know, the macaque brain is about 10 or 15% the size of the human, so it's not the same, but it's closest as opposed to, oh, I don't know, say a mouse, which is 1/3000th the size of the human. So that's okay. And so with Elisa Brewer and Stelios Smirnakis and Nikos, you know, we went over there and we could use the same methods to go and find, and the macaque, this is the operculum, they call it back here, this is the eccentricity map. And this is one of the reasons why the macaque is such an attractive model, because V1 is just on the surface right back there. So it's easy to get at with microelectrode and so forth. And so we could go and do that. And Stelios, Stelios is a neurologist and was a postdoc or something. He had some position with Nikos at the time. He's now at Harvard. And he could lesion the left eye and the right eye in the corresponding locations. And that would therefore remove the inputs to this chunk of V1. And so that gave us a controlled situation where we thought, okay, great, now we're going to see it plastically reorganized. We're going to do some biochemistry and look at the molecular basis for this and so forth. And we measured, LPZ stands for lesion projection zone. So he had lesioned the two eyes, and we measured right away, and then we measured 2 weeks later, and then we measured 4 months later, and then we measured a year later, not shown here. And you know, the thing just didn't close up at all. Just didn't close up at all. And this really ran in the face of lots of other, Charlie Gilbert and so many other people were saying, oh my God, there's all kinds of plasticity. But they were finding their plasticity by going in with anatomical measures of boutons or looking a little bit of sprouting. So what they were seeing were things that were maybe 50 microns in size. And we were looking at something here that was many millimeters across. And it just, these are just plots, you know, time after time. And plus we were looking at fMRI and they're looking at anatomical. So, okay, maybe it's electrophysiology, but you know, it's Nikos's shop, and Nikos went in with the electrophysiology, and he came out, he would come out, I remember one time in particular, he came out, Nikos is a very Greek, enthusiastic, vigorous guy, and he's only about this big, but he can still shake quite well, and he said, it's the deadest piece of cortex I've ever measured, you know, that kind of thing. So we just weren't seeing it. And there's a little bit of residual conflict in the literature. This happened to get published, accepted for whatever reason in Nature. And it's a little bit of back and forth about that. And I don't think we convinced them. And I know we've seen this again and again now. We saw it, I didn't want to keep flying to Germany and working on this thing with monkeys. It's a project. It's complicated for various reasons. But I did have the good fortune to work with Yuichiro Masuda and Hiroshi Horiguchi, who I had a series of wonderful ophthalmologists from Japan who came by and would work with me on projects like this. And they really led projects on other kinds of retinal lesions, natural ones that you guys probably study and treat routinely, JMD, Leber's, we had Leber's patients, retinitis pigmentosa patients, and they have now published a series of articles, and they've been great. I helped them, you know, find the coffee and get started on this work and shared some tools, but they have really pursued this. And the same thing has been true in all of these kinds of human subjects, that when in the face of macular degeneration, it's not the case that cortex changes and fills in, and there's a lot of plasticity. And again, we're sticking with our story on this, but you know, it's a pushing and shoving going on a little bit in the literature on that. Okay, so I will now tell you a case study in this general thing. Perhaps Some of you will have heard of the famous Cheselden case. If not, it's just kind of fun. Is this showing up here? Oh, good, it's not. The Cheselden case, where it was the first case really where somebody had very bad optics, very bad physiological optics. And Cheselden did a surgery in 1728 and hoping that the kid that he did the surgery on would be able to see, and the kid didn't really see very well. Richard Gregory had done a remarkable case of when they first did the corneal transplants in London in the 50s. Richard was a brilliant guy and went rushing down to the hospital to see, well, what happens? This person had been blind for, I think SB had been blind for many years, and now all of a sudden the cornea has been replaced and he has a clear retinal image. And what does he see? And nothing very good happened in that case. And Sachs tells him a similar story. Valvo tells a similar story. And we had a case like that for the first time. All of those were behavioral cases, right? These were corneal replacements that you could then study their behavior and ask them what they see. And that's a great thing about studying humans. You learn a lot. But this guy here, Mike May, and Mike is very public. He's written books about it and so forth. Nothing secret about his case. Mike had who had lost vision. One eye was just completely blown out in a chemical and thermal explosion in his parents' garage when he was about 3 years old. But one eye was still intact, but the cornea and the limbal stem cells were completely blown out. And every time they tried to put in a new cornea, it would just cloud up. And they tried that once and it didn't work, and they didn't want to do it anymore. And so he lived for 43 years with one eye very, very clouded. So he had no contrast. He could, if the room lights would turn on and off, he could see that, but he couldn't see any patterns or structures. It was so crowded. He was quite a remarkable guy. I'll tell you more about him in any event. When he then went in, maybe you know this person, it was a Dr. Goldman in San Francisco, whom I don't know, but who learned how to do these corneal replacement with the stem cell therapy. And here's a picture of Mike just coming out as it's just taken off the bandage. He's reaching out to touch his then wife. First he'd ever kind of seen her face and so forth. And we became, and Ione Fine, who really got into this, met Mike and started testing him and followed him up quite a bit. And she called me up one day and said, so like, you know how to do that MR thing, and we don't really have any MR data on people like this. Brian, how about if we all get together and we do some magnetic resonance imaging of Mike to track it? And she had been doing, and we helped with some just standard tuning curves. And maybe you guys know the spatial frequency on this axis. So how many cycles per degree on a pattern. And this is Mike May's data at various times, various months after the surgery. And you can see he can see stuff at very low spatial frequencies. And 0.25 is very low for a spatial frequency. You can all see out to 30, 40 cycles per degree. The controls in this region are all flattened here. And Mike starts to fall off like this. And so even after the surgery, after the replacement, after he has a good retinal image, after he has a good ERG, electroretinogram, he still has very poor spatial resolution. And I happened to 84 months after the original surgery, I happened to be driving past Davis where he lived, and I stopped in and took another measurement. Just didn't change, just didn't move much. And Mike, by the way, even though he had this surgery, and he doesn't regret it, and he could sort of see stuff that he couldn't see before, some amount of patterns and so forth. He still had a seeing eye dog, and he had a cane, and he lived his life, and he's quite a remarkable guy. He's an entrepreneur, and he just had all these rigs and stuff, and he kind of lived his life not that differently after the surgery compared to before the surgery. So that suggests that all those years living without any contrast vision meant something had happened to his visual cortex that just wasn't going to get fixed after the corneal replacement. Okay, so it's a kind of stability. 43 years of this, sure, I'll give you a good retinal image now, but you know what? Even though for these achiasmics, the cortex has no problem. Come in developmentally, I can figure out anything. Give me this, give me that. I'm okay, I'm good to go. But for Mike, after 43 years, Cortex, now all of a sudden an image comes in there and Cortex says, I'm sorry, I'm cooked. You know, I'm ready to go. I'm not changing. So there was a couple of things. I'll show you the responses for Mike. This is this temporal occipital region here. When you put in a moving stimulus that really, you know, flickers and moves and so forth, you'll always get a really big response in temporal occipital cortex here. Sometimes maybe you'll have heard of MT. It's kind of a famous visual area that's very filled with motion responsive cells. And he actually, he had. This is a control here. You can see, I think it's actually Elisa. And you can see that moving stimulus produces all kinds of activity. In Mike, it wasn't very much, but MT was there. We could see that. Mike, by the way, as I mentioned, he was special. Mike, as a blind guy, competed in the Paralympics in the skiing event. And for quite a number of years, he held the world's record for downhill skiing for a blind guy. And the speed was 100 kilometers per hour. And if you wonder how a blind guy skis at that pace, it's you get the world gold medalist to ski in front of you, and he tells you, go right, go left, stuff like that. He leads you down over the moguls and stuff like that. So Mike was, that's why, you know, he's out there with a book, and he was hoping that Robert Redford would play him in a movie and stuff like that. So, so he had a lot of flickering lights and lots of kinds of things, you know, transitioning across his visual field. And so perhaps that kept us alive. When Netta Levine, who's now in Jerusalem and is chair of the neurology department there, I was fortunate to have her in the lab for a couple of years. And when we compared controls, you've seen this before from fovea to periphery, in Mike, all that foveal stuff, all that red stuff, just, you know, we just never could see a signal like that. And we could, in fact, this is the sizes of the PRFs. I was showing you these things that they go up, like in typicals, they go up, up, up like this. In Mike, they're bigger and then they come down. In fact, the size he has here in the periphery isn't that different from the size of these guys, the typicals in the periphery. But in this fovea, the size is way too big, consistent with his inability to see fine patterns. Okay, and things like face areas and stuff is just, you know, we don't get that out of Mike. We did also look at the tractography in his case, and he is off the distribution. So Mike is the star over here. This is longitudinal diffusivity and radial diffusivity of the optic tract. And Mike here's the, you know, the ellipsoid is the usual, cortical is the distribution. We also compared him to blind and monocular patients and so forth. So something did happen from the age of 3 on to the white matter in his brain that was quite different. And the corpus callosum was a really big effect. Here's Mike. He has a very big corpus callosum, but a very small crossing compared to all of these others. It's just diagnostic. I wish I had a computational model of what implication that would have. And if you know this, what would that mean? And what should we expect? And what could we do? I'm sorry, just a little aside. I wanted to say that I have just become amazed at the last 10 or 15 years about the white matter and the axons in the myelin. I'm just kind of shocked by how much we've learned. So maybe you all know this, but the axons in the middle of the white matter turn out to be releasing BDNF, brain-derived neurotrophic factor, and vesicles, for God's sake, containing glutamate and GABA that actually excite the glial cells. So I'm just sort of thinking, boy, I should spend more time. Well, when you're younger, maybe you'll spend more time looking at what the impacts are here. In any event, all of this plasticity and ability to think about plasticity led us to the conclusion the way we summarized it. Stelios and I wrote a review article some number of years ago, and we said, you know, asking whether the brain is plastic or stable is just a waste of time. It's both. depends when and where. It's how I started. I'm so glad that you're nodding. So it's a little comfort because sometimes I say that and people kind of actually get annoyed with me. And I just think we need to figure that out. And obviously during development, there's a lot of plasticity. And so I then became interested at that point in time. We had sort of done enough on the maps and we thought, well, maybe we could do something helpful for kids and look at the development of the visual circuitry that was part of, that was necessary for reading. And I have to say, when I got into that field, and I would encourage you all to have a look and see if there's something you can do in there also. There's just a ton of kids who, not mine, but a lot of, friends, kids, and people where the kids just have trouble reading, and there's an obvious why. And it's not really a corneal effect. It's not a lens effect. It's nothing obvious about the retina, but there's something going on. So that kids, some people who can be very successful in many other ways, just can't learn to read, just a skill that they don't have. Famous people, maybe you see them routinely. Charles Schwab go around giving talks about this and start centers for this. His kid can't read, but yet Schwab is a successful guy, and David Boies, I don't know, all kinds of people. So we thought, well, let's try to do something. To see if we can't figure this out, something to at least contribute to this. And like any complex failure like that, failure to learn something as complex as that, it's not likely that there's one single bottleneck. So we sort of laid out, Rosemary Lee and I laid out in a review article, all the places that we should start checking. And to just sort of get you to where we were, I should say that I should either remind you or explain to you that on the ventral surface of the human brain, there are several regions that seem like they're highly specialized for parsing, for understanding the foveal signals. So actually what comes down here is not the entire visual field a lot, but mostly the fovea and regions right around it. A lot of the axons from the foveal retina, if you track where they actually end up delivering their signal, it's into the ventral occipital regions like this. And people had things like this, achromatopsia, if you have lesions over here, Meadows in 74 said, you know what, your color vision can really become problematic. And if you have lesions in these red zones over here, maybe it's more likely to be you have difficulty seeing faces. So there's kind of a big industry of people, and I'm not one of them, but I watch them, who have gone and said, there's a face region here or something like that. But I'm not also a person who looks into a single region and says, now that I know that this is responsive to faces, I understand faces. I'm a kind of person that, well, how did it get there? And where's it going to? And what's the computations and stuff like that? So we thought, well, let's see if we could do that. And this is from my colleague, Connie Grill-Spector, where it's showing some of the complexity, the review from about 6 or 10 years ago, showing some of the complexity even at that time, and it's just gotten more. So we know where these maps are. V1, V2, V3, we're looking at the ventral surface here, and there had been a region that Stan DeHaan and Laurent Cohen had identified as being particularly responsive when you see words, see words. Not quite the same as reading, just seeing words. And so they called that, they had a lot of confidence. They called it the visual word form area. Might do other things, I'll say, but it does respond when you look at a word. And with Andreas Rauschecker, who's now at UCSF, we, in the neurology department there, we could put on a word. So you're fixated here, and you put on a word. Okay, it's in the right hemifield. So you all know that therefore it goes into the left hemisphere, and it's just out there on the little periphery. So there's, you can see the response in V1. And you can also see the response in V2. And V3 right next to one another over there. This is the horizontal meridian. So it should be along the border there. And sure enough, there it is. There's another map down here, the fourth one. Then there's another, these ventral occipital ones that I showed you in the beginning. And then there it is, VWFA. So for me, that was good. That meant we could start to try to trace these signals at least that far. And maybe if we got better, we could keep going and find other regions. These are the same data I'm just showing them two different views of the left hemisphere, the one I tracked for you. I've just rotated around so that you could see that on the dorsal surface, there's some activity, it just doesn't go quite as far. And so we just started making measurements. And I told you about these population receptive fields that Serge and Kaoru had been making with me for a number of years. And in those regions here, where I was showing you down here in the VWFA, you don't have a map, but you do have individual voxels that for each one of those, they have a receptive field. And even though they're not a map, and so you can take them and plot them all next to one another. They're kind of a little scrambly, you know, you can't see a clear map, but you can put them up and say, well, how much of the visual field is covered? How much does it see? And, you know, if you do that in the early ones, you know, which we did just to confirm that this all made sense, you'd say, well, V1 covers a hemifield. Cool. Okay. And then V2 and V3, when you go down here, covers a quarter field. So, okay, what does it cover when you get down here? It's not a map, but how much does it cover? And Gary and I still owe a paper on this, and Rosemary Lee, this is part of her dissertation. And you can see that, wow, this is only 5 degrees over here. And for this particular one of 20 subjects, you really only have a small coverage. And so that the regions that are responsible for interpreting words don't really see out to here. And as Zig will tell you, if you put letters and words out in the far periphery, you can't actually read them very well. Try it yourself. You know, just look at the exit sign, then look off the side and ask yourself whether you can see. And there's a number of reasons why you can't read it when it goes out there. And one of them, we think, is because the signals that you learn to read with go down here, and they only go into the ventral region. They only come from the fovea, and that's what you train on and so forth. And so when you have that signal in the periphery, it doesn't get down there, and the parts of the brain it goes to are not trained for seeing words. That's what we think. And we started looking at different people. So the 20th subject was the one down here, and they're not all the same. Some, you know, some are similar and some are varying and so forth. And this is kind of, we have one more story to tell. I won't tell you about this today, but you know, we looked at these and thought, well, maybe this could be a diagnostic cue about who's going to read well or not. But we kind of stopped because, you know, you can compensate with eye movements. So we started to try to measure for the ones that have small regions and bigger regions, they have differential eye movements and so forth. And there's a piece by Jesse Gomez thinking that maybe they do. So we were kind of in that mode where, okay, we have these measurements, we have some eye movement measurements, but we pretty much around 2018, 2020, we sort of stopped in COVID and plus on hold. Okay, so what have I been doing now in my retirement after that? So we've got this story up to reading a certain amount. And I'm telling you the story about building it, the signals up from the beginning. And I have to say that I felt that starting at V1 with the signals, as I did for you in that previous map, wasn't good enough. And plus, I just like hanging out with Palanker and the ophthalmologists and, you know, people who do physiological optics things and cameras and so forth. And so Dave Brainerd, Nicholas Guitarras, Trisha Leanne, and I, a number of others, we said, you know, I don't want to do another $50,000 experiment. So we've opened up our computers and we started trying to build computational models to summarize all of these data, starting at the beginning, at the stimulus, and going all the way through the optics, through the retina, and so forth, and build those things. And with the idea that, you know, we've got these measurements down here of what's happening in ventral cortex. We've got ways of predicting them. We know things, by the way, about the white matter that I didn't tell you about, but there's another dozen papers about thinking about, we've discovered a new track, and anyways, a bunch of things in the white matter. So we'd like to assemble it all into some kind of an actual, here's the stimulus, here's what happens through the lens, here's what happens in the retina, here's what should happen in V1. let's just go. Let's try to build this up, right? And why should the computer scientists have all the big computers? Why couldn't the ophthalmologists have some big computers? I don't know. We ought to be able to do that. So we ought to be able to write our code in a nice, transparent way, share it, have digital twins of the visual system, so that when a patient comes in, you have like what the standard should be at different places. And here's a little computational model of that and so forth. And if they have trouble reading, maybe you look at the digital twin and look at their measurements and try to understand why. So it's a good way to retire. You can do this in coffee shops and so forth. And we have open source code image systems. The image systems engineering tools were things from that other part of my life having to do with cameras and CMOS imagers and so forth. But these have now been repurposed. So we have parallel extensions into trying to do the front end, take the Pablo Artal measurements of the wavefront aberrations, and we incorporate those in all things that are known about the inhomogeneities of the retina and so forth. And we're building these up, and we hope they'll be useful to you. And I hope I'll have enough time to be able to use them in thinking about how it is letters and words and so forth get carried on through the visual system through these cases while kids are learning to read. So let me say that the work that I described to you has been work over quite a number of decades. Not all of it was ours to begin with, but the work that I described was carried out by many of these people. Here's Dave Brainerd as a young guy. I don't know if you know who this person is as a young guy. This is Elisa, was very helpful. E.J. Chichelniski is Daniel's colleague at Stanford in neurosurgery. Bob Doherty was very, very helpful. Heidi Bassler did a lot of the early work with me on plasticity and so forth. Tony Morrill and Alex Wade are at York University and doing great. John Winnower continues to be a close partner of mine. He's a wonderful colleague at NYU, Ariel Rokem, Kendrick, so forth. I particularly want to mention these guys here, who a lot of the work on plasticity, and particularly the case studies that I showed to you, came from the fact that I was just fortunate, right? A lot of this, you just have to be lucky. Like, you have a great place here and people are going to want to come and visit with you and take advantage of the fact that you're in such a beautiful part of the world and such nice buildings and so many smart people. And I was fortunate that way. And these guys from Tokyo just called me up and said, look, we're ophthalmologists in Tokyo, but we kind of want to get PhDs, and why they want to do that, I have no idea. But I was just great, because we give out PhDs and you know a lot of stuff. So that's great. And they came, and it was a wonderful, probably decade of these ophthalmologists teaching me about Leber's and macular degeneration and doing patient studies and so forth, which let me talk about quite a number of things that we shared here. So thank you all very much for your attention, and nice to meet you.
So before we go to Gordon, he has a question on Zoom.
Okay.
Let me ask you, you mentioned about 16 different maps or regions that you identified. Did you do a color stimulation as well?
Yes.
Can you comment on that? I just don't know the literature.
Yeah, sure. Sure. There was a period, I'd say it was in the late 90s, maybe in the 90s and early 2000s, when Samir Zeki was, really pushing the idea, following on Meadows, that there was a color center of the brain. And he had an important publication with Friston, where they would put up a target was black and white, and then another target of the same pattern, but that was colorful. And they would find one place that was heavily activated for that. And that was in the days when we had weak, our tools weren't that great, and you'd see one thing pop out. But if you do that experiment now with much more sensitivity, you'll see that those very same data produce differential activations all kinds of places, not just the one place. It's a thing about having an insensitive instrument means that maybe you only pick out the one really big one, and then the others are just below, and then your instrument gets more sensitive. My God, it was like everywhere. So the idea that there's a single color center, particularly for somebody like me, who I spent like a quarter of my life worrying about the cone photoreceptors and photopigments and stuff like that. There's a series of things that lead to your color and lead to your color perception. Although Samir, whom I love dearly, was really focused on the idea that there is a color center and that your experience of color is all tied to that. It's definitely, you know, if you screw up your cones, your experience of color is screwed up. If you screw up the certain populations of ganglion cells, your experience of color is screwed up. If you screw up this part of the visual cortex, your experience of color is screwed up. So it's part of the chain.
I was more even specific. If you have, let's say, deficiency in blue cones, short wavelength cones, whether there's a different organization on this mapping versus, let's say, those who have red and green deficiency.
That's interesting. We did different things than that. I don't have the maps won't change. I haven't measured that, but the maps are not going to change with respect to that. But we have looked to see where the S-cone signals go. So the S-cone signals, as many of you know, come out through the bistratified ganglion cells into the coniocellular layers of the LGN, and then they go into the superficial part of V1 instead of into they have a different path. And then they may or may not go to other parts that we didn't know about. So we particularly looked to see if we could get S-cone driven signals in motion selective cortex and so forth. We did that for a couple of years, and it did seem that their distribution was different compared to the LNM codes. So that part, you know, not all parts of cortex have the same color responsiveness.
Gordon.
Yeah, hi, Brian. This was an absolutely wonderful talk. I just learned so much and so incredibly clear. And I expected it to be a wonderful talk because I've heard your talks previously and they've all been wonderful. About a month ago, Pawan Sinha from MIT gave a seminar at a symposium the NIH organized on plasticity. And he's been working, as I'm sure you know very well, he's been working on these kids in India with congenital cataracts who've had their cataracts removed, and is asking questions similar to the ones that you've been asking about your patient, Mike.
Yes.
So that raised two questions in my mind. First of all, in Mike, did you see any auditory compensation? Did you look for that? And then what are your thoughts about the effect of the age on the extent of plasticity?
Yes. Thank you, Gordon. Thank you so much for the kind words. So Pavan has led this project, it's probably been 10, 15 years, called Project Prakash. And he has done it's really a combination of science and humanitarian work in India, where he will persuade people like you who do corneal surgeries and so forth to go help these kids. A lot of them have these corneal lesions because their moms don't have, what's the disease? It's like they don't have the MMR thing, and so they get measles, mumps, rubella, I guess. So they get, so that when they're born with this disease that then scars their corneas. And if you had enough money, but in lots of parts of India they don't, you can replace these corneas and give these kids sight. But a lot of them grow up 10, 15, 20 years. And like Mike May, where their corneas are not in good shape. And so they go in and they do these surgeries, and Pavan has published a couple of papers, but Gordon, I haven't seen any papers recently, but he did publish papers maybe 10 years ago on collections of these kids. And sometimes when they do these surgeries at the age of 15 or 20, actually the behavior gets quite a bit better. And that wasn't true for Mike. And sometimes it doesn't get any better. And we don't have enough, I would say, power on knowing the individual cases. And, it's in India and it's remote and they don't do neuroimaging on it. And the behavioral measures are important. They're navigational measures mostly. Can these kids navigate? And so I down pathways, and maybe he showed you a few others. They had some, they used to make occasional resolution measurements. Did he show that in his last presentation to you?
So what I remember is that there was improvement in many of these kids. It took a long time that they initially did not see very well, but with time they definitely got better. And he also had evidence for auditory compensation. That's why I really asked the question.
Yeah, okay.
I haven't read any of his papers. I just heard this talk, which really impressed me.
Yeah.
Perhaps he's got a lot of data. I don't know.
So I will say that you say auditory compensation. There have been reports also that if you don't have good visual signal, I'm not sure it's compensation, but that you can pick up either memory or auditory or non-visual signals in regions that are normally largely visual. I want to say just one more thing. I went to one of Pavan's talk, and I really admire his efforts here. So I don't, I went, actually, it was with this guy, Hiroshi Horiguchi, and we went kind of enthusiastic about this effort. And he came out of there as a clinician, and he said, you know, the cost of doing these surgeries and catching these kids at this point in time compared to the cost of giving the proper pills to the moms so that the kids aren't born with MMR. So it's like $1 for a pill. And the cost of the surgery is thousands of bucks. And the number of, so if you were just looking at social policy, there's nothing wrong with what Pavan does. But if I was a legislator in India and I wanted to spend $1 million on something, I would be spending it on the pills, not on the surgeries. So I just wanted to add that. I think Pavan is also trying to get some science out of it. And the science has been, you know, slow in coming over the last 20 years.
So Brian, we have a challenge here. We have 12 or 13 questions and we have 10 minutes.
I'll be shorter. I'm sorry.
We need to move on. And
sorry.
So either that or we'll reduce the number of questions.
I'll be shorter.
Okay, so I will be next. Thank you very much for this fantastic presentation. And going back to your subject that didn't have the chiasm, but you mentioned that he had a good vision, but he also had some degree of small nystagmus.
He did.
Was there any correlation between the two eyes in the nystagmus?
Yes, they did. They moved together.
Very interesting. So do you think this was to help him? He didn't have the 3D vision, but maybe that was to help him with the 3D reception? What is your...
He saw in 3D, but he had no stereo.
Yeah.
No stereo.
Thank you.
Thank you. That was really fascinating work, and I really appreciated the historical perspective. Gordon kind of asked my question. I remember reading a long time ago about people who lost their sight and kind of repurposing the visual cortex for other senses. So can you comment a little bit on that more? Is it only in congenital blindness, or is it also seen in people who lose their sight at some older age?
Right. The big reports are the initial one was from Sedato in the late 90s, and they're pretty much congenital are the big ones. Ones at a later age, maybe they squeak out a little signal, but not a big effect.
Thank you.
Thank you.
So, detailed question perhaps. When you described the early visual areas V1, V2, V3, and you showed the topographical maps, Eric Schwartz was describing them using the log polar map as a geometry. Now, when I spoke to Schwartz, he was quite strong that it exists in V1, V2, V3, but it was not clear how far it goes beyond those three. Did you look into the log polar nature of the higher visual areas?
I didn't really. There have been people, so I didn't, it was a bit of a quantifying the maps in terms of an abstract theoretical representation was a passion of Eric's. But I would say that other people looked at it and said, yeah, it's a good approximation, and here's 4 other good approximations. The data are sufficiently noisy. The maps, I'm going to say it's a fine approximation, one of many.
Hi, beautiful talk. Sometimes, pharmacology allows us to have beautiful tools to study, for example, your maps. And I want to know, if you actually ever use any of these molecules that have very funny visual effects and see what happens to the maps of these healthy volunteers.
Absolutely not. And make sure you tell all my funding agencies that I would never do such a thing. But on the other hand, that was really why we set up the whole rig I think it's a great thing to do. And that was why we set up the thing in the Max Planck Institute. Yeah.
Okay, we have two questions from Zoom. Brian?
Hi, from one Brian to another. I've always been sort of fascinated by the brain's intrinsic plasticity and the ability to process new information. And when the Knightses did their experiment where they engineered new opsin into primates to give them, dichromacy to trichromacy, the cool thing for me was not the fact that they could do that, but that the brain was like pre-wired to see the additional discriminant and make sense of that. Do you think that that's like new recruitment of additional cortex, or this is kind of a riff on the earlier question. But I'm just wondering about your thoughts on that.
Thank you. For everybody else, the Knightses have a study where they took dichromatic, I guess in their case, it was dichromatic monkeys, right, macaques, and yes, engineered one of the pigments to shift it a little bit. And the monkey could then make discriminations that were not possible prior to this engineering thing. And there's a bit of a battle in the literature, and I don't think it's resolved, which is why I went to explain this, as to whether the reason that the monkeys could then do the new discrimination was literally because they had they had now trichromatic color vision, or it was because the introduction of the opsin, and we've computed this, produces a kind of speckle pattern in the image. If you had you could do those discriminations if you had no new color, just because you'd have a kind of a textury new image. And Walt Mack pointed this out first, and we did a lot of calculations on it, and I'm not sure that's resolved. The Knightses are convinced, and perhaps you are, that actually this really we were ready for a new dimension of color, and they're pursuing this with Austin Rohrder and others at Berkeley to give all of you not 3 dimensions of color, but 4, not 4, but 5. And so this is an ongoing conversation. Put me in the skeptical camp, but I'm very respectful of the Knightses. They've done tremendous work, and maybe they have something, and I'll think, yeah, you really could have 5 dimensions of color.
Regardless of whether it's an additional color or not, it's an additional sensory modality that they've been engineered in.
No, it really could be just you look at something and it looks spatially uniform, and you look at something and it's got a lot of little speckly light and dark things in it, and say, yeah, I can tell the difference. It really doesn't have to be. So literally I can do those experiments computationally, right, without any new invoking any new experience. But I think come and visit us and talk about it. I think it's a terrific topic.
I'd love to. Yeah.
All right, let's move to Chair.
Hi, great talk. Thank you, Brian. My question really is about, did you look at the effect of deliberate learning, like things that Daphne Bellevere has done, Dennis Levi, Robert Hess?
I tried to hire Daphne, but no, I've never gone into things. Yeah, things like the word attention didn't come up, or intentional and so forth. I'm not that far advanced, but I love those guys, and I don't have much to say about them. We do have a guy, Jason Yateman, at our place in the Graduate School of Education, who probably does do things like that, and Justin Gardner, who you might enjoy visiting.
Thank you.
Hello, thank you for the great talk. I'm just wondering if you have had a chance to look at the brain from people with albinism. They have a reduction in IPSI and more contralateral projections. there could be some problems because in terms of the fovea acuity, but I was just wondering.
I missed a key word there. But for autistic kids, is that what you said?
Albinos.
Oh, albinos. Let me refer you to Tony Moreland, Morland. Maybe you already know his work, but he has spent a decade. Tony was in my lab. He came wanting to work on albinos. He didn't, but he left, and he has done tons of work. Michael Hoffman, kind of that crowd. And yes, they have reported a lot of things, but maybe if you haven't seen it, that's a great place to start.
Thank you. Thank you for the reference.
Hi, Dr. Wandell, over here in the room. I have a question about how the reading circuitry may be affected in patients with aphasia such as Broca's or Wernicke's. Are they still able to read?
I just think that's a great, right? So then it's the next stage on. And I have not gotten to that, and I think it's important. But I'm hoping, for example, if you do it, that you do it by taking the systematic approach all the way through and not just looking for a spot somewhere. But I have not. But thanks for the question.
Hi, again in the room. I really enjoyed your talk, and I understood one thing from your presentation. Correct me if I'm wrong. That so-called critical period is longer than up to 3 years of human life, since it wasn't able to be repaired after cornea transplant. That's one question that I like the confirmation. And the second is, do you know what happens in aging actually? Whether there is a loss of precision or somewhat problems, I don't know, amplify from retina to cortex or the opposite. Thank you.
Yeah, thanks. The question not only of what is, I think if we break the critical period up, I think that's a consequence of maybe something we both agree on, that some things are fixed a little earlier than others. So I didn't mention about Mike, and it's been true of all the patients, that they see color instantly. So Mike was good till 3, and at 43, he still saw color just fine. Other stuff. At 3, he probably could see out to 25 cycles per degree. But the development of acuity seems to continue a lot longer than color, so that by the time, when it got screwed up for all those years, he lost the acuity, but he kept the color. So probably we want to probe him into different subsystems like that. And I have not done work on aging, but in fact, when I look around me, I'm noticing that all the people in the center, I've now stopped directing that center, and just a few months ago, and all the people are working on infants and kids, and I keep saying, well, what about me, people? I'm 72. You could put old people in there, but in the places I've seen, I haven't seen a lot of elderly studies.
All right, we will conclude with the 3 questions.
Okay. Thank you for this presentation. I was wondering if you ever work with amblyopia in terms of like this. It also seems like a very good model for this plasticity versus the stability. Have you ever worked with amblyopes? And just a quick other question. In the case of stroke or lesion in V1, do you see upstream effects on the retina?
Oh, yeah, okay. I have not. It's a great topic. And sometimes I give people references. I'm sure people have worked on amblyopia, at least strabismic amblyopia. I'm not sure about NISOs and so forth. But I have not, and I'm sorry, I can't even give you a reference on it. But I know it's out there. And as per the lesions, what we did look at was not functional stuff, but we did look at whether there was white matter impacts projecting back, and I can't. And I'll look it up and I'll send it, but I don't have a clear memory of it.
Hi, great. Yeah, I really enjoyed that talk, just like everybody else. So in the visual word finding area, is the activity contingent upon recognition of the characters? So if you put, Chinese characters or blurred characters in English, would the activity be the same?
Yeah, that's thank you. And that's very interesting. In China, there are several groups literally asking whether, because their reading and writing system is so different, whether in fact they even have a visual word form area. And the general view is yes. But then as to whether it's the same or not became important to them because of surgeons who go in and do brain surgery and don't want to, and they want to check whether the reading system for the Chinese characters is the same. It's not. And there's a debate between a group down in Shenzhen and a group up in Beijing, and I don't know how it's going to turn out. But they do have the original one near visual cortex. Everybody agrees on that. And whether the secondary ones are in the same place or not, and how the surgeon should act, was not resolved last I checked.
So Brian, now I'm giving you Zach.
Thank you for that really interesting talk. I was really curious, has anyone ever looked at synesthetics and whether there are interesting changes in like VOT fields of synesthetics, particularly if they have these kind of very hardwired connotations between like words or letters and colors, for example?
Yeah, thank you. So first off, thank you for that. Synesthesia, there's some wonderful behavioral studies on synesthesia. We have Eagleman hanging around Stanford. For some reason, he did a ton of synesthesia stuff. There have been people who looked to see whether white matter connections in synesthetes were different from controls. And I'm sorry, I don't have on the top of my mind who that was, but I'm guessing you could find, you look for tractography and synesthesia, but I would also guess it's not that sharp a measurement so that you could actually see a few.
Right, and I imagine it's kind of dependent on the individual.
Yeah, but behaviorally, there's still a lot to be learned from synesthesia. And the fact that it's a very real phenomena, not just some kind of goofy thing, is quite compelling, actually. And Randolph Blake, I notice, is winning an award at VSS this year, and he's done wonderful work on synesthesia.
Thank you.
Thank you very much, Brian. It was fantastic.
Thanks, everyone.